Hello, today we'll be talking about a question that came in GATE 2023 Biotechnology paper. Which of the following methods are used for detection of DNA and RNA respectively? Southern and northern blotting, southern and western blotting, northern and southern blotting, northern and western blotting. So we'll discuss a little bit about the blotting techniques and what do they mean and what will be the answer of this question. So this is a blotting compass. In this schematic you can see which technique is what. Basically the northern blotting, southern blotting and the western blotting are the most frequently used techniques. The others are just modifications that are there that you can also work with in the lab. So what is blotting basically? Blots are nothing but techniques for transferring DNA, RNA, nucleic acids and proteins onto a carrier so that they can be separated and often follow the use of a gel electrophoresis. The southern blot is used for transferring DNA, the northern blot for RNA and the western blot for protein. So different blots are used to identify the presence of one specific target molecule in a complex mixture of related molecules. Okay, and it basically, the blotting technique refers to the transfer of macromolecules from a gel onto a solid surface of an immobilized membrane for the detection of the transferred molecules. And all blotting techniques share a similar workflow. Initially, an electrophoretic procedure is used to separate molecules by size on a gel based on the movement of macromolecules in electric field, and then these separated molecules are transferred to a solid membrane. It can be nitrocellulose or nylon PVDF suited to immobilize the target molecule, and then end users can then incorporate labels, radio label or fluorescent label, to the immobilized targets with probes that are sequence specific or shape specific to the target molecule of interest during an incubation step that facilitates hybridization. And the hybridized complex, which consists of a bound probe, can be later detected and visualized using various imaging methods. Okay, so one of the techniques is southern blotting. A little bit on the history. Southern blotting was originally introduced by Edwin Southern in 1975. It's an analytical technique in molecular biology research that end users use to basically measure the size and amount of specific DNA sequences in a complex mixture through immobilization of the target sequence to a solid support, followed by hybridization of a complementary DNA probe. Shortly after southern blotting was developed, basically the premise of this analytical technique was applied to the measurement of the size and amount of RNA transcripts from a gene of interest, which is known as northern blotting. Both blotting methods share a similar workflow: sample preparation of purified high quality DNA or RNA from a sample, then electrophoresis to separate nucleic acid fragments by size, then transfer or blotting to a solid support that immobilizes isolated target nucleic acids, then preparation and hybridization of nucleic acid probes, and lastly the detection of the nucleic acid probe. Okay, so these are the three main types of blotting techniques that are very frequently done in most molecular biology labs. So to talk about a little bit about the southern blotting technique. So during sample preparation, both southern and northern blots begin very similarly. The target DNA or RNA is purified from samples through nucleic acid extraction, and purified whole genomic DNA or plasmid DNA or RNA transcripts are isolated using common extraction techniques already optimized. After extraction, the southern and northern blot protocols begin to differ. Okay, southern blotting requires an additional step because of the need to take purified DNA and partially digest into smaller different sized double-stranded fragments with restriction enzymes that cut the DNA at specific spots. So to explain the technique a little bit, this method involves separation, transfer and then hybridization. Okay, and it is used to detect the presence of a particular piece of DNA. And this DNA can be a single gene or it can be a part of a larger piece of DNA such as the viral genome. And the key to this method is hybridization. Okay, the process of forming a double-stranded DNA molecule between a single-stranded DNA probe and a single-stranded target DNA on the membrane. Okay, so the mixture of molecules is separated on a gel electrophoresis system. As we know, in a gel electrophoresis system, the sample can be loaded, and based on size it will be distinguished, because it will migrate from the negative end to the positive end. The smaller molecules will go much faster, will travel greater distance than the larger molecules, which will go very slowly. Okay, so that is how the mixture of molecules can be separated. Then the molecules can be immobilized on a matrix that can be transferred to a solid support blot. Now gels are not that stable and they can diffuse very easily, which is why for the next part of the study all these molecules are then transferred to a solid support. Okay, then the labeled probe, so we know the kind of the DNA of interest that we want to detect. Okay, so we can then synthesize or commercially purchase a probe that is complementary to that sequence of DNA, and it can be labeled. It can be a radio label or fluorescent label, whatever. It has to be a labeled probe that will give a readout basically, and this labeled probe can then be added to the membrane to bind to the molecule. Unbound probes will be removed obviously by a washing step, and the place where the probe will be connected will attach to the target of interest, DNA of interest. It will correspond to the location of the immobilized target molecule. Okay, so the DNA to be analyzed, such as the total DNA of an organism, is extracted via optimized protocols and then digested to completion with restriction enzymes. We all know restriction enzymes are basically endonucleases that have specific nucleotides where they cut. Okay, so for example, EcoR1, HIN3, all these are very popular restriction enzymes used in molecular biology labs. Now that they have been digested into smaller parts, the complex mixture of fragments is then separated via gel electrophoresis to separate the fragment according to size. Smaller fragments will travel faster than larger fragments, which will take a lot of time to travel. The restriction fragments are then present in the gel will be denatured with heat or alkali and transferred onto nitrocellulose or nylon membrane by blotting. And this procedure, basically what happens, a replica of the gel will form on the membrane. Membranes are much more stable, there will be lesser chances of diffusion happening there. So a replica of the gel will form on your membrane. So wherever your DNA of interest was, it will be at the exact spot on the membrane as well. The filter will then be, the membrane will then be incubated under hybridization conditions with a specific radio labeled DNA probe. So the DNA probe that you will commercially purchase or synthesize will have a radio labeled to it or a fluorescent label, and that will basically lead to the hybridization process between the probe and the complementary DNA fragment, DNA restriction fragment. The excess probe, which will also attach due to non-specific interactions, will be washed away, and the probe bound to the filter will be detected by autoradiography, which reveals the DNA fragment to which the probe has hybridized. Okay. So this is the basic schematic of the technique. The DNA, its restriction digested, separated on a gel, transferred to a membrane. Then membrane bands are hybridized with the radio label, and then bound DNA bands are external film. So as you can see, this was my DNA of interest in different, these are different samples. This is my particular DNA of interest. Okay, so once the gel has done, it has been transferred to the membrane. Now the probe, this probe that we have taken from our side, will have a sequence that will be complementary to this sequence. So when I'm adding it to this membrane, it will go and bind to the specific regions only. There might be non-specific interactions, but that will be taken care of during the washing step. Okay, and then when I'll expose it on a film, I'll be able to see at which part the DNA has come up. So this is what the hybridization will lead to. So for DNA, the probe can be made of 20 or 30 nucleotides. It will be a complementary to your region, and there are two methods of labeling. As I said, radioactive labeling or non-radioactive like biotin. So radio labeling is very sensitive, but it also hazardous because obviously radioactivity. Non-radioactive labeling is sensitive also, but it is relatively much more expensive. So this is the basic concept of hybridization, the binding between single-stranded labeled probe to complementary nucleotide sequence in the target DNA. Okay, and detection of hybridization depends on the method of probe labeling. So these are the steps I've already talked about: digestion into DNA fragments, size separation, then in situ denaturation. Okay, and then transfer of the denatured DNA fragments into a solid support membrane, so as to because the gels are much more likely to diffuse faster, the bands on the gel are likely to diffuse faster. Then the hybridization with our labeled probe, detection, and then estimation of the size and the number of bands generated, the detection and the analysis afterwards. So there are different methods of transfer that can be used. These are very popular methods of transfer from the gel to the membrane. All of these have been studied, and it has to be optimized according to the experiment that someone is working on. So obviously Southern blots are very useful in gene discovery, gene mapping, evolution and development studies, and in regard to genetically modified organisms, Southern blot is used for testing to ensure that the particular section of DNA has been successfully incorporated. So you know that this particular section of DNA I want to incorporate into the particular genome of the genetically modified organism or GMO. So you can then synthesize a labeled probe that is complementary to the particular size of a particular genetic sequence, and then you can do the hybridization analysis and do Southern blot to find out. Also, detection of mutations are also very can be performed using Southern blotting. So any a mutation in only one nucleotide will change the restriction endonuclease recognition sequence, and when the restriction endonuclease will fail to recognize and cleave at that site. Okay, So the presence of a mutation that will affect a restriction site will cause a pattern of bands that will differ from those with a normal gene. So a normal person will have the normal wild type sequence. A person who has a particular disease or particular mutation will, even if one nucleotide is different, that nucleotide might be in a restriction endonuclease recognition site, and if that changes, then the restriction endonuclease no longer recognizes that sequence and it will fail to cut there. So the pattern of bands will be different for the control person and the diseased person. So that is how people might figure out that is one way to figure out the detect mutations and basically figure out if a person has a particular disease or not. The northern blotting process is quite similar. As I said, it is the technique for detection of specific RNA sequences. It was developed by these two, George Stark and James Halvin at Stanford University, and was named such by analogy to southern blotting. Okay, so RNA is isolated from several biological samples. The first step will be that. Also, RNA is much more susceptible to degradation than DNA. Whenever someone is working with RNA, you have to be much more careful and much more patient because RNA degrades very fast. Okay, and then the samples are loaded on gel, and the RNA can be separated also according to their size, just like the other nucleic acid DNA, and the resulting gel can be seen. It will be based on the size of the nucleic acid. Then it can be blotted onto a nylon membrane or a nitrocellulose filter paper by creating a sandwich arrangement. Obviously, basically a replica of the gel will be formed on the membrane, as you can see from this image over here. And then the image can be placed in a system where the synthesized or the purchased probe complementary to the RNA sequence of interest will be added, and there will be multiple washing steps after that, and then to remove the unbound probe, and then the labeled probe can be detected via audio radiography or via chemiluminescence reaction. In both cases, this will result in the formation of a dark band on an X-ray film. So you can find out in which sample the probe, the RNA of interest is in, based on how the probe has hybridized to that particular position. The probe added has hybridized to this position, to this position, to this position. That means these three things. This will be a visual readout as to at which parts, which bands the probe has hybridized to. Okay, the applications are one and many. It's a standard study for direct study of gene expression at the level of mRNA, study DNA degradation, RNA degradation, RNA splicing, RNA half-life. All of these can be done. Often it is used to confirm and check transgenic or knockout animals as well. Another blotting technique which is very, very frequently used in most molecular biology lab is Western blot. So in this case, the only difference is that the analyte is protein. It's not a nucleic acid, it's a protein. So the protein of, in case of proteins, we use STS page or page electrophoresis, polyacrylamide gel electrophoresis. And after the proteins have been separated according to size, after you have narrowed down the proteins according to size, you can then transfer the pattern of protein bands onto a membrane, a compatible membrane, and then do the similar steps. Instead of the probe in this case will be an antibody. Previously we were talking about nucleic acid, we were talking about a complementary sequence probe, but here, since we are detecting a protein, an analyte of interest is a protein, we have a primary antibody specific to that particular antigen. Then we can add a secondary antibody conjugated to an enzyme. To that, the secondary antibody is specific to our primary antibody. It will attach to here and it will lead to signal amplification. And then if we add a substrate of interest, substrate of this enzyme, then this enzyme will convert this substrate to a visual readout. So more the quantity of protein in a sample, more binding of the primary antibody to the enzyme, more binding of the secondary antibody to the primary antibody, which has the enzyme conjugated to it, more conversion of the substrate to its colored product, and more is the visual readout. That is how we can find out the presence of a particular protein of interest in our sample. So the question that we started off with was, which of the following methods are used for detection of DNA and RNA? It's southern blotting and northern blotting. Option A, DNA is detected by southern blotting, RNA by northern blotting. Western blotting is used for protein detection. If you want to learn more about this blotting technique and how they are used, how they are performed in labs, you can go to this course, Genetic Engineering Theory and Application by Dr. Vishal Trivedi, IIT Guwahati, and you can see these YouTube videos for more information. Thank you.